Good morning, everyone. I am Jayson Pandoy, and in this video, I will be discussing the first part of Chapter 19 in Microbiology, which talks about diseases of the immune system. So, these are our learning objectives, and these are the topics that are covered under this chapter. And for my part, I will only be discussing about hypersensitivities. The other topics will be discussed by my groupmates in separate videos. So, to start our discussion about hypersensitivities, here is a question that you may ponder on. What is an allergic reaction? So, an allergic reaction is an immune response to a type of antigen called an allergen. When we say antigen, these are indicators or markers found on certain substances that tells our immune system whether they are harmful to our body or not. And allergens are specific type of antigen. They can be found in many different items, and unlike other kinds of antigens, allergens are not necessarily associated with pathogenic microbes. So example of allergen-containing products are peanuts, milk, eggs, and seafoods like shrimps and crabs. And as you can observe from these examples, they're actually not harmful to the body, but some individuals are sensitive or allergic to them because their immune system recognizes the allergens found in these products as harmful to the body, and they trigger an immune response, which is called an allergic reaction. So next, let's talk about hypersensitivities. So as we all know, we have an immune system that protects our body from pathogens and harmful substances that may make us sick. But sometimes our immune system can also inappropriately react or exaggerately react to certain stimulus, which can lead to undesirable reactions or complications called hypersensitivities. So, allergy or allergic reactions are an example of hypersensitivities. And now let's talk about the types of hypersensitivities. We have four types. We have type 1, type 2, type 3, and type 4 hypersensitivities. So for the first type, it is a rapid immune response that occurs almost immediately when a pre-sensitized individual is exposed to an allergen, which means that type 1 hypersensitivity is just similar to allergy, or all allergic reactions are type 1 hypersensitivities. And the immune reactant or the primary antibody that is at play in type 1 hypersensitivities is the IgE antibody or the immunoglobulin E antibody. This antibody sensitizes an individual to type 1 hypersensitivity reaction upon subsequent exposure. So, once an individual is sensitized to type 1 hypersensitivity, once they are exposed again to a certain allergen, then allergic reaction would occur. So let's dig deeper into the process of type 1 hypersensitivity with this picture. So as you can see on the first part, upon first exposure to allergen, APC processes antigen and presents it to Th2 cell. So this is the APC or the antigen presenting cell. It engulfs the antigen, processes it, and then presents it to the Th2 cell. So once the allergen is presented to the Th2 cell or the T helper 2 cell, the T helper 2 cell will release IL-4 and IL-13, which activate B cell. So IL-4 and IL-13 are cytokines, which are small signaling proteins that regulates immune response by allowing the immune cells to communicate with each other. So Th2 cells and B cells are immune cells, and they need to send signals in a form of cytokines to ensure a smooth immune response whenever there are a presence of antigen. So once the B cells are activated, they would proliferate and differentiate into plasma cells that synthesize and secrete IgE antibody. So as you can see, the B cells right here, differentiated into plasma cells, which produces IgE antibodies. Now, the IgE antibodies binds to mast cells by Fc region, sensitizing the mast cell. So, the mast cells are another immune cells which contains chemicals that are responsible for the signs and symptoms of allergy whenever there is an allergic reaction. So in the first exposure or initial exposure, the IgE would only bind to the mast cell to sensitize them or to activate them, making them ready for the next exposure. And upon subsequent exposure to allergen, mast cells with IgE bind to antigen and release inflammatory molecules resulting in allergy symptoms. So after the initial exposure, if the individual is exposed to the allergen again, the allergens would bind to the IgE antibodies, and as a result of that, the mast cells will release the chemicals that they contain, and these chemicals lead to allergy symptoms like inflammation of the skin. So these are the chemicals that are contained within mast cells. And as I mentioned, these chemicals are mostly responsible for the allergy symptoms and signs that is observed whenever an allergic individual is exposed to allergen. So we have heparin, histamine, and serotonin. And these are common examples of type 1 hypersensitivities or common allergies. So the first one is the allergy induced asthma. Second is anaphylaxis. Third is hay fever. And the last one is hives or urticaria. So all of these allergies are localized allergies, which means that they can only be found or the signs and symptoms are only visible on localized body parts. But for anaphylaxis, it's a systemic reaction to allergens, meaning its signs and symptoms can be found in other parts of our body, even if only a localized part of our body or the localized part of an allergic individual's body was exposed to the allergen. So now let us proceed with type 2 hypersensitivities. Type 2 hypersensitivities are also called cytotoxic hypersensitivity. They are mediated by IgG and IgM antibodies binding to cell surface antigens or matrix associated antigens on basement membranes. So remember, on type 1 hypersensitivity or allergy, the primary antibody is the IgE antibody. But for type 2, the primary antibody is the IgG and IgM antibodies. And in type 1 hypersensitivity or in allergy, the antibody, which is the IgE antibody, only binds to mast cell, which results to the release of chemicals that leads to allergy symptoms like inflammation. But for type 2 hypersensitivity, the antibodies, which are the IgG and IgM antibodies, binds to cell surface antigens or matrix associated antigens or antigens that are found on the extracellular matrix. And as I mentioned earlier, type 2 hypersensitivities are also called cytotoxic because they lead to the death or the lysis of the targeted cells. And the antibodies, IgG and IgM antibodies, can either activate complement or they can be involved in antibody dependent cell mediated cytotoxicity or ADCC with cytotoxic cells. So when we say complement, these are proteins found in cells that aids the immune system in or during immune response. So when there's a presence of harmful substance in our body and the complement proteins are activated, it can lead to a cascade of reactions in order to eliminate those harmful substances. And it can result to the death or cell lysis. And as mentioned right here, it also involves cytotoxic T cells. So cytotoxic T cells also leads to the death of the cell because they are specifically or especially created to eliminate the affected or the targeted cell. So these are common examples of type 2 hypersensitivities. We have hemolytic disease of the newborn or HDN, and we also have hemolytic transfusion reactions or HTR. So before we dive deeper into the common examples of type 2 hypersensitivities, let us first learn about ABO blood group incompatibility. So ABO blood group, I am confident that you are already familiar with this concept because this was already discussed in our previous course, the genetics course. But yeah, I will only be providing a short overview about the ABO blood group. So in the early 1900s, Karl Steiner first described that individuals have different blood types. So we have blood type A, blood type B, blood type AB, and blood type O. So the ABO blood type is determined by the presence or the absence of surface carbohydrates on our cells, which are the A antigen and B antigen. So if there's a presence of A antigen on the red blood cells, then the individual is a type A person and they have anti-B antibodies on their red blood cells. And if there's a presence of B antigens on their red blood cells, then the individual is classified as a type B person and they have anti-A antibodies on their red blood cells. Now, if a person has both A and B antigens on their cell surface, then they are classified as type AB individual and they don't have any antibodies on their red blood cells. And if a person does not have any surface carbohydrates on their red blood cells or they don't have both A and B antigen, then they are considered as or classified as type O person and they have both anti-A and anti-B antibodies on their red blood cells. Now, it is very important that we know the blood type of an individual, particularly in the medical field, to avoid any complications during blood transfusion. So, before blood transfusion, it has to be ensured that the blood type of the receiver or the recipient is known and the blood that will be transfused to him or her is compatible with his or her own blood type. So, as I mentioned earlier, one of the examples of type 2 hypersensitivities is the hemolytic transfusion reactions or HTR. So, this disease is it happens when a blood recipient receives a blood that is incompatible with his or her own blood type. So in this example, the donor is type A and the recipient is type B. So the donated type A blood with type A antigens enters bloodstream of the type B recipient and then the anti-A antibodies and plasma of type B recipient bind to donated type A red blood cells. So since the anti-A antigens or anti-A antibodies recognizes the type A red blood cells that enter the bloodstream as harmful, then they would attack these cells by binding to them. Bound anti-A antibodies activate complement cascade, causing hemolysis and release of hemoglobin. So the type A red blood cells would be destroyed by the anti-A antibodies of the recipient, which can lead to the death of the red blood cell or what we call the hemolysis and the hemoglobin will be released. And once this happens, it can lead to what we call coagulation, which can cause kidney failure or possibly death of the recipient. So another important thing to remember during blood transfusion is the RH factors. RH factors are the second most frequently described RBC antigens, or antigens that are found on red blood cells. They were also identified by Karl Landsteiner, same with the ABO blood group, but this time he is with Alexander Weiner in 1940. The RH system of RBC antigens is the most complex and immunogenic blood group system. When we say immunogenic, it means that a certain substance is can highly trigger an immune response. So RH factors can highly trigger an immune response, especially if it's incompatible or if it's recognized by the immune system as harmful. Now, if a person is RH positive, that means that the raw or the antigen is present on the red blood cells. And if a person is RH negative, that means that they don't have raw or D antigens on their red blood cells. And RH factor antigens are proteins. So earlier in ABO blood group, the A and B antigens are carbohydrates. But for RH factors, they are proteins. So another example of the type 2 hypersensitivities, which involves RH factors, is the hemolytic disease of the newborn, or HDN. So on the first part of the picture, it shows a pregnant mother with a RH positive fetus inside her. So the mother is an RH negative person, which means that both their blood are incompatible with each other. So the red blood cells of the RH positive baby can enter the mother's bloodstream. And since the mother is RH negative, the immune system of the mother can trigger an immune response, which leads to the release of anti RH antibodies. However, for the first pregnancy with the RH positive plus fetus, the baby is unaffected because the baby would already be born before the mother's immune system can produce enough anti-RH antigens to attack the baby's red blood cells. So for the first pregnancy, the baby is safe. However, for the second pregnancy with the RH positive baby with the same mother, um, it can now lead to the hemolytic disease of the newborn, or HDN, because the anti-RH antibodies that were produced by the mother's immune system during the first pregnancy is already circulating the mother's bloodstream. So once the baby is conceived, the anti-RH antibodies would then enter the placenta and then attack the baby's red blood cells. So that leads to the hemolytic disease of the newborn, which is fatal to the baby and could possibly lead to the baby's death. Now, the other part of the picture shows a first pregnancy with the RH positive fetus and anti-RH antibody treatment. So there's a treatment to avoid hemolytic disease of the newborn for second pregnancy with RH positive plus fetus. On the first part of the treatment, the mother is injected with raw or the immunoglobulin or immune globulin during pregnancy. So this is done so that once the baby's RH positive red blood cells enter the mother's bloodstream, the injected raw or the immune globulin would release the anti-RH antibodies that are contained within them to eliminate the RH positive red blood cells from the baby. And in this way, the mother's primary immune response would not be activated. Because if it's activated, then it can lead to the the accumulation of the anti-RH antibodies on the mother's uh, bloodstream, and once the mother gets pregnant again with an RH positive baby, then those anti-RH antibodies that are already present on her bloodstream would attack the baby. Now we're done with the type 2 hypersensitivities. Let's talk about the type 3 hypersensitivities. So type 3 hypersensitivities are immune complex reactions that were first characterized by Nicholas Maurice Arthus. Immune complexes are an excess of antibodies coupled with a relatively low concentration of antigen. Immune complexes, these are like clumps of blood that is consist of antibodies, primarily IgG or IgM antibodies that are binded to antigens with which have relatively low concentration compared to the antibodies. So meaning there are more antibodies compared to the number of antigens. So the type 3 hypersensitivity is classified as immune complex reaction because this reaction or this hypersensitivity can lead to the formation or the accumulation of immune complexes in the body. So the immune complex accumulation leads to a cascade of inflammatory events that include the following. So first is IgG binding to antibody receptors on localized mast cells, resulting in mast cell degranulation. So this is similar with allergies or type 1 hypersensitivity, wherein the antibody binds to the mast cell and sensitizes them. And after the subsequent exposure, it results to the release of the chemicals contained within the mast cells, which are responsible for the allergy symptoms. For the second one, complement activation with production of pro-inflammatory C3A and C5A. So as I mentioned earlier, complements of these are proteins that aids our immune system in fighting off harmful substances. So the C3A and C5A are examples or specific types of complement proteins. C3A specializes in increasing the permeability of our bloodstream, which means that substances can easily go in and out of the bloodstream. And C5A, it recruits the effector cells or immune cells that fight off harmful substances in our body, like macrophages and neutrophils. An example of type 3 hypersensitivity is the serum sickness, which is a systemic type 3 hypersensitivity. And as I mentioned, when we say systemic, it's not constrained to a localized part of our body, but can be found in other parts of our body as well. So as you can see on the picture, this is the immune complex, and as you can observe, there is a relatively high concentration of antibody compared to the concentration of antigens. And these are the organs or body parts that are affected by the accumulation of immune complexes. We have blood vessels, heart, joints, skin, and kidneys. Now let us proceed with type 4 hypersensitivities. Type 4 hypersensitivities are regulated by T cells and involved the action of effector cells. So unlike the other types of hypersensitivities, the type 1, type 2, and type 3 hypersensitivities, which are all mediated by antibodies, type 4 hypersensitivity is directly mediated by immune cells, which are the T cells and the effector cells. So T cells, these are responsible for recognizing and remembering antigens in our body. We can think of them as the commanders of the immune response. And effector cells, these are the cells that directly fights or immediately fights the harmful substances in our body once after being recognized by the T cells. So we can think of effector cells as the soldiers. So T cells are the commanders, while effector cells are the soldiers of the immune response. So these types of hypersensitivities can be organized into three subcategories based on T cell subtype, type of antigen, and resulting effector mechanism. A specific example of type 4 hypersensitivity, which is classified under the subcategory 3, is contact dermatitis, which is a result of exposure to allergens from poison ivy, which is called the hapten. So, in this example, on the first exposure or the sensitization, the allergens enters the body, and then the T cell, which are the commander responsible for recognizing and remembering the antigens, they would bind to the antigen, and then once they bind with the antigen, the T cell would release cytokines, the small signaling proteins that I mentioned earlier, which regulates the immune response by allowing immune cells to communicate with each other. The release of cytokines would send signals to other immune cells like the macrophage and cytotoxic T cell to fight off the harmful substance that is present on the body or the antigen. And this reaction can lead to the formation of lesion or lesions on the body. And this can significantly damage the tissue of the skin. Now we're done with talking about the types of hypersensitivities. Um, Now we have another concept called the hypersensitivity pneumonitis or HP. It is a disease caused by more than one type of hypersensitivities. So a combination of types of hypersensitivities. This disease goes by different names associated with various forms of exposure. We have fancier's lung, poultry worker's lung, cheese handler's disease, farmer's lung, sauna taker's disease, and hot tub lung. So, um, these names of hypersensitivity pneumonitis are based on the setting or the environment where the individual acquired the disease. Now, let's talk about diagnosis of hypersensitivities. We have two methods of diagnosing hypersensitivities. First one is the prick puncture skin test or PPST, and second one is the intradermal test. So the PPST is the most convenient and least expensive way to diagnose allergies, and it is carried out with the introduction of allergens in a series of superficial skin pricks on the patient's back or arms. And for intradermal test, it requires an injection into the dermis with a tuberculin needle attached to a syringe containing a small amount of allergen. So compared to the PPST, which only involves superficial skin or shallow skin pricks, the intradermal test goes deep into the skin. Both the PPST and intradermal test are observed for 15 to 20 minutes for a wheal flare reaction to the allergens. Measurement of any wheal or a raise or itchy bump and flare or redness within minutes indicates a type 1 hypersensitivity. So the PPST and intradermal test are only used for diagnosing type 1 hypersensitivity or allergies. Other types of hypersensitivities cannot be diagnosed using these methods. Now, let's talk about the treatments of hypersensitivities. So, prevention of allergic reactions from insect stings and other environmental allergens can be achieved by desensitization or hyposensitization therapy, which reduces the hypersensitivity reaction through repeated injections of allergens. It makes the immune system of the individual get used or tolerate the presence of antigen or allergen in the body. And other treatment of milder allergic reactions typically involves antihistamines or anti-inflammatory drugs and analgesics. So these are mostly used to treat allergies once the allergy reaction already occurred. Preventing further exposure to the antigen is also necessary. So, as the saying goes, prevention is better than cure. So it's necessary that once you know that you are allergic to a certain substance, then you avoid taking in or being exposed to that substance to avoid hypersensitivity. So that would be all for hypersensitivities. Just a short recap. We have four types of hypersensitivities: the type 1, type 2, type 3, and type 4 hypersensitivities. For type 1, the primary antibody at play is the IgE antibody, and type 1 hypersensitivity is just allergy. For type 2, the primary antibodies at play are the IgG and IgM antibodies, similar with type 3. And for type 4 hypersensitivities, it is unique among all the types of hypersensitivities because type 4 hypersensitivities are not mediated by antibodies. Rather, they are mediated directly by immune cells, specifically the T cells and effector cells. So that will be all. Thank you so much for watching this video. You can check the videos of my other groupmates for the rest of the topics covered under the diseases of the immune system. Thank you.